I'm still a little bit nervous speaking to this audience because I am not a computer scientist. I'm a poor biologist trying to speak the language, so please bear with me if I talk too much about biology or if I make some naive comments on computation. At the beginning, I want to introduce a very basic concept in molecular evolution, which sets the foundation of my method. The concept is called sequence conservation across species. When we study a gene in human, we can choose to look at that gene just in human, or we can choose to look at a gene across different species. Here I have human, but I also listed several other species, and these 8 species span 500 million years in evolution. If I'm interested in one gene, I would retrieve the sequence of that gene from all species that are available and align them. In this picture, each row represents a sequence from a species, and each column represents a residue at a position. This fragment showed us this particular gene is pretty conserved over the time because they look very similar in different species. If I pull out a different gene, you can see this one has the freedom to change. The question is, why we have these 2 different patterns? The answer is the nature action on them. If a mutation occurred and the mutation affects a position or a gene that is functionally very important, the individual that carry that mutation will not survive. So over the time, that mutation will be purified from the system. On the other hand, if a mutation occurred on a less important position, in a less important gene, the nature doesn't care, so it stayed. And over the time, it may be lost or it may be fixed. So we will see genes with different levels of conservation. The first case represents the highly conserved genes, and behind it, it means this gene is essential to the survival of the cell or the individual. And the second case represents a variable gene, which means you can change in sequence, you can also change in expression level, and the individual may not be affected. We have different ways to measure such conservation. One simple way is we measure the rate of evolution. This is as simple as just to count for each position the different number of residues and then divide it by the evolutionary time span in the unit of number of substitutions per site per billion years. And notice this is by position. So if we have a protein, we can measure the evolutionary rate of a protein by simply average the rate over all positions. If a protein evolves fast, it actually means it's less conserved. So to use the evolutionary rate as a conservation score, I have to take the reciprocal of that. Okay. In the last slides, we covered different species and the mutations and the sequence changes occurred on the germline level, which means it can be passed to its descendant. Cancers are different. They're somatic events, which means the mutations are not passed to your next generation. So in one of my research interests is to see if this somatic event also follows some long-term evolutionary patterns. And surprisingly, somatic events in cancer also reflect germline patterns. In this picture, I showed 3 groups of genes. One is oncogene, the other is tumor suppressor gene, and the third one is just any gene, random genes from human proteome. And we can tell oncogenes is among the most conserved genes in whole human proteome, followed by tumor suppressor genes. And furthermore, if we find cancer driver mutations occurred in oncogenes, they occur on highly conserved site. On the other hand, if we just sample human population, those are healthy people. They carry mutations on fast-evolving site. And on the expression level, I do not have the result for cancer data, but people have done analysis and experiment in yeast. They reported the expression of conserved genes is also under greater constraints as compared to variable genes. And all this conservation comes to one thing, which is if the changes affect conserved genes, then the functional implication is more severe compared to the changes that affect variable genes. This knowledge, when I use them in the modeling, translate into a very simple idea, which is I would like to give higher weight to proteins that are more conserved. But instead of using 8 species, I use 46 species. And cumulatively, these 46 species cover 2 billion years of evolutionary time in the history. So this should be a pretty thorough coverage. I calculated the conservation score for all 231 proteins, and here's the distribution of their conservation score. As you can see, majority of them are not conserved, but there is a subset of genes that are particularly conserved over the time. The most conserved one has less than 0.5 substitution per billion years, which means this gene just cannot tolerate any changes. And there are also 40 clinical features for which I cannot calculate the evolutionary rate, so I just simply give them the highest weight among all proteins, which is 19.6. The other weight I use is a measurement to measure how significant they are differentially expressed between the two classes. And I used a log p-value from the student's test. And the distribution of this negative log p-value is as expected. Most of them are not differentially distributed, and a small subset of them are more has a significance. And this is the differential weight. And I combine the evolutionary weight and the differential weight to give me a final weight for each feature. So next, I'm going to talk step-by-step process in my model. First, I dummy code the categorical variables, and then for missing data, I use random sampling to replace their value. And then I removed features that has low information content, and there's only 4 such features. I also try to detect redundant features that share high pairwise correlation, and I couldn't find any. And it turned out the organizer has already removed them, so this is not a surprise. And then I took a very, very simple transformation to normalize the scores, which is the G transformation. And then the Z scores is multiplied by their corresponding weight. In the training data set, I found they are not balanced because the number of samples with complete response is almost 3 times as many as the number of samples that show resistance. So the first thing I do is I do an undersampling to generate a subset which contains the equal number of samples from both classes. And then based on this subsample, I performed the feature selection step. And this feature selection was performed on weighted values, which is the Z-score multiplied by the weight. For the feature selection, it's done through sparse logistic regression with stability selection. The stability selection is to handle different regularization parameters that may affect the number of features to be selected. I did 100 bootstrapping for 20 regularization parameters, and those features that were selected in more than 50% of bootstraps were used in the downstream classifier building. To build the classifier, I removed the weight. So the classifier was built upon z-scores without weighting, and this is done from a random forest algorithm. And after this, I would have a random forest classifier. And this subsampling, feature selection, and the classifier training step was repeated 100 times. So at the end, I have 100 random forests. And they composed of an ensemble model. For a given sample, I would run that sample through all 100 classifiers and count how many of those models predict this particular sample to have a complete response. And the confidence score is simply the percentage of those models giving complete response prediction. The performance of this approach has a balanced accuracy about 78% and area under the ROC curve about 0.8. And then I got curious about how those model, what features goes into this model and what are the weight and so on and so forth. So I first look at how many features each model include. Overall, each model includes about 30-some features, but some models may have features as few as 19 or as many as 41, but on average, this could account for relatively less complex models. And then I sort all those features based on the number of models they were used. Out of those 231 proteomic data and proteomic features and 40 clinical features, only 81 of them was included in at least one model. The rest of them were never used in any model. And interestingly, all of them are proteomic data. Some clinical features may be used in fewer models, some may be used in more models, but shows clinical features do have a higher probability to be included into those models. And some proteomics data simply has no information at all. So after 100 rounds of iterations, they still provide no information. And then I look at the top few features that are most frequently used in more than 80% of models. And I found, and I listed them, there are 8 of them. The top 6 are all clinical features. And the 7th and the 8th are proteomic feature. And we can see the 7th is PIK3CA. This is a well-known oncogene. And the evolutionary weight is 9. And let's see where it's located in the distribution. It's located on the right tail of the distribution. So it shows this PIK3CA indeed is extraordinarily conserved genes. This GSK-AB, unfortunately, I was not able to find a human protein with exact name. So I cannot compute the evolutionary weight accurately. So I asked the algorithm to just randomly assign it, and for some reason it assigned it with the highest evolutionary weight. But later I got curious. I tried to see if there's anything I can do to pinpoint what this protein is. And I suspect this is a GSK3B gene. And GSK3B gene indeed is highly conserved. It's somewhere around here. It's even more conserved in PIK3CA. And then I examined the differential weight. All those 8 features has p-values pretty low, and especially the top one, it has a p-value of negative 10 to the power of negative 6. So the differential distribution is really strong. So the conclusion is evolutionary information can actually help us to identify genetics features that are functionally more important. In my research, I studied evolutionary patterns for disease mutations for quite some time. For a long time, we suspect that evolution can inform some practice in diagnosis or prognosis. And because we can compute the evolutionary conservation not just on protein level, but on positions, so we can do similar things not just for protein expression, but also on SNP data. We can do position by position evolutionary weighting on all kinds of genetics information. And also we can compute protein coding positions and non-coding positions. We can also code for regulatory elements. So we sort of becomes very excited after this challenge to find out, at least we know coding and expression data. With this particular data set, evolution information really helps. There are certain things I should have done, but didn't have time because I know about this challenge about 3 weeks before its closing date. So I have to hurriedly put together a method and then submit it. I was lucky to find out it works well, but at the same time, I didn't have the luxury to tune my models to try different parameters or algorithms. For example, I probably should not use the random sampling for imputation. Then I can avoid some confusions. And also, since I'm not a computer scientist, I can now integrate this evolutionary information into the modeling in a more sensible way. For example, people has mentioned to me after this challenge that I should have integrated the information in a penalty term of something, okay? And he wrote down an equation to me, and I look at it. It's just a school of tadpoles, so I just I just clueless about what I can or cannot do. What I have done is very, very intuitive, but fortunately it worked. So on one side, it shows the limitation of biologists trying to pretend to do computation work. On the other side, it shows if we do have some collaboration, it may open a door to some unknown possibility. And I have a special thanks to Dr. Jieping Ye. He is the computation scientist who showed me the equation. And he also the person who developed the SLAP package. Dr. Ye and I has been collaborated for 4 years on several projects. And I use his SLAP package for any machine learning related tasks. And I really appreciate it. If I understand how to use this SLAP package, I can say, he really does a good job. Okay, and the last thing is I want to thank the organizer of the Dream Challenge. I don't know what other people think, but for me, this is a very unique chance because, as I said, we suspected evolution have some usage in computation. But we never was able to find something that can actually test our hypothesis. This is the first try we did, and it turned out to be so good, and I have to give a lot of credibility to the organizers. Thank you.